Welcome everyone to Nanoscope's clinical perspective discussion, which features presentation and discussion of Nanoscope's lead clinical candidate MCO10, an ambient light activable multi-characteristic opsin MCO, optogenetic monotherapy, and its potential role in treating retinal degenerative diseases, including retinitis pigmentosa, which we'll refer to as RP, and Stargardt disease, two inherited retinal diseases which together represent the leading cause of blindness in working age population. MCO10 has been granted orphan drug designation by the FDA for both diseases. On a personal note, I'm deeply honored to be part of this team that is working together to develop an effective treatment for RP and Stargardt disease. As someone who has relatives who have been affected by RP, I have personally seen the devastating impact this causes to patients and families. Any product that restores vision would therefore reverse the course of natural history and make a huge difference to patients' lives. RP is a rare genetic disease, and unfortunately, most patients eventually lose most of their eyesight. There are approximately 90,000 patients in the US that are blind due to RP. Stargardt disease is the most common inherited single gene retinal disease, characterized by macular degeneration that begins in childhood, adolescence, or adulthood, resulting in progressive loss of vision. Roughly 15,000 patients in the US are blind due to Stargardt disease. Currently, there are no cure for either disease. However, we believe that Nanoscope's MCO is the only therapeutic platform that provides broadband light sensitivity with fast kinetics and ambient light activation for vision restoration. Let me show how this technology works in this video presentation. In the case of RP and Stargardt, the photoreceptor cells responsible for normal light perception degrade. Nanoscope MCO therapy imparts light sensitivity to bipolar cells, which are abundant and retained after disease-related photoreceptor loss. In this way, MCO therapy makes bipolar cells act as new photoreceptor, taking the place of those lost to disease. Nanoscope's MCO10 gene therapy utilizes intravitreal injection, a convenient and well-established office procedure to deliver an AAV packaged transgene encoding the ambient light-sensitive MCO protein into retinal bipolar cell. This leads to robust and long-lasting expression in bipolar cells, which persist after photoreceptor loss. A large in number, also uniformly organized, providing the potential for high-quality vision restoration. Because the bipolar cell layer is naturally integrated with the existing visual processing circuitry of retina, light-sensitive bipolar cells are able to efficiently pass visually evoked signal to the brain through the optic nerve and thus restore vision. The MCO molecule is sensitive to broadband light, meaning these visually evoked signals are generated in MCO-treated patient in ambient light environment. Nanoscope's MCO optogenetic gene platform has potential to address all forms of retinal degeneration and is applicable for patients suffering from complete or partial damage to retina. We are developing MCO10 with the goal of changing the treatment paradigm and providing vision restoration to hundreds of thousands of patients. Over the next 12 to 18 months, we anticipate several potential value-creating milestones. In the first quarter of 2023, we expect phase 2b3 top-line data for MCO10 in RP and phase 2 6-month data for MCO10 in Stargardt. In the first half of 2023, we plan to conduct an end-of-phase meeting with FDA for MCO10 in RP to align on requirement for the BLA submission. In the early 2024, we anticipate that we'll submit a BLA for MCO10 in RP and expect an approval and commercial launch around the end of 2024. Now let me turn our agenda for today's event. We are very fortunate to have two distinguished guests with us today, Dr. Stephen Sang and Dr. Victor Gonzalez. Dr. Sang is an acclaimed geneticist in patients with retinal degenerations and a practicing ophthalmologist at the Columbia University Irving Medical Center, Edward Harkness Eye Institute in New York City, and specializes in medical retina. Dr. Gonzalez is a practicing ophthalmologist and is the founder and CEO of Valley Retina Institute, McAllen, Texas. Dr. Gonzalez is also affiliated with Valley Baptist Medical Center, Harlingen, South Texas Health System, Harlingen Medical Center, Mission Regional Medical Center, Rio Grande Regional Hospital, Knapp Medical Center, and Doctors Hospital at Renaissance. Dr. Gonzalez is also co-investigator in one of our clinical sites for Stargardt and RP studies. From the Nanoscope team, Dr. Samar Mohanty, our co-founder and president, will detail the molecule and design of MCO10 and review preclinical data amassed today. Dr. Aaron Osborne, our CMO and CDO, will review the clinical trial design for our fully enrolled phase 2B program for MCO10 in RP and our phase 2 fully enrolled multicenter trial for Stargardt disease. Aaron will also moderate an interactive panel discussion and will address your questions. Before we get started, I want to state that neither Dr. Sang or Dr. Gonzalez are employees of Nanoscope, but they have been compensated for the time in relation to this event. And all opinion expressed during this discussion belong to them and do not necessarily reflect the opinion of their affiliated institution. Now, let me turn the call over to Dr. Stephen Sang. Dr. Sang.
Thank you, Sawatna, for giving me the opportunity to share with you the status of my field and also some of the work done in my laboratory. So we'll focus on retinitis pigmentosa today. Retinal degenerations affects about 10 million people, and retinitis pigmentosa is the most common form of inherited form of retinal degeneration. Patient initially lose the rod, the night seeing cells, and then eventually lose the cone cells, leading to this eventual central vision loss. To talk about the impact of retinitis pigmentosa, I'd like to bring you the attention of how CMS looked at payment. How do you compare cataract surgery from gallbladder surgery? Your quality of life 1 means perfect health. Quality of life 0 means death. When a patient cannot see 20/20, which is the perfect vision, they would have a quality of life similar to someone with an amputation or chest pain. You need 20/40 to pass the driving test in most states. When patients get down to about 20/60, the quality of life units that patient will fill out will be the same as someone that is on dialysis or hip fracture. When patient cannot see the big E on the eye chart, the big E means about 2400, so it's pretty close to 250. The cost taking care of a blind person would take at least $47,000 in 2006 value. In terms of thinking about treatment for retinitis pigmentosa, there's a staging system done by Jose Sahel, Burton Rasker, and Jean Bennett. At stage 1, when the cells are still there, so patient need to be fairly young, that you can see both the rods and cone cells are present. And then gene therapy, giving the gene using genome surgery will be highly effective. And this is how the strategy used in the first gene therapy approval in medicine by Medicare and Medicaid. When then the rods are gone, then the cones are left. There's a lot of trial that's been used using neurotrophic factor or antioxidants. There's a current phase 3 trial. Eventually, the cones become sick and die, which is at a later stage, stage 3. And then many pharmaceuticals is using optogenetics to kind of make these remaining cones more robust by introducing channel rhodopsin for the cones. And at a later stage, then when eventually all the cones are gone, you still have some bipolar cells left and the ganglion cells. So electronic retina that can stimulate the ganglion cell, or stem cell replacement to replace those died photoreceptors has been used. The challenge for gene-specific treatment, precision medicine, there are about 80 different genes can cause retinitis pigmentosa. May not be so practical to treat all 80 different genes by going to FDA 80 different times. And also the conventional treatment is using subretinal injection. Patient need to go to the operating room to inject underneath the retina. As you hear, some of the optogenetics is doing intravitreal injection, which can be done in the office. It's less traumatic to the patient. This is an example in my laboratory of a subretinal injection in a retinitis pigmentosa mouse model. After a single treatment, you can see that all these mottling salt and pepper retinopathy are gone. One single treatment for the lifetime. These are one of my patients now getting the FDA approval Luxturna therapy. Before treatment, the patient doesn't do vitamin A metabolism, and that's why it's black. And after treatment, it restores vitamin A metabolism, then you can see that the autofluorescent returned. And patient improved by 3 lines of vision. Although this precision medicine treatment has been highly successful, the cost maybe make it less practical. There's already reviews written that maybe you should think about in precision medicine treatment. For example, cystic fibrosis have a highly effective treatment for 1 mutation. This drug only works for 1 mutation in the gene cystic fibrosis. And it costs $300,000 per year. It's interesting how they price a drug for $300,000 per year, because before the FDA approval, the treatment for a cystic fibrosis patient is about $300,000 lifetime. Patient for cystic fibrosis before this new drug, unfortunately, would die from bronchiectasis or pancreatitis in the 40s. But if precision medicine become available, say for Stargardt, it will cost about $30 billion. And Medicare has only about 700 billion. So there's lots of editorial written about the cost of precision medicine and maybe need to consider imprecision medicine. And now I show you that optogenetics is one of the tools to this imprecision medicine to treat all these different 80 genes in patients with retinitis pigmentosa. Stage 1 needs to be gene specific and cells need to be present. And stage 2, there's some trophic factors, all including antioxidants. And I'll show you some examples of that in the next few slides. And when all the cells are gone, then cell replacement stem cell is a prosthesis electronic retina, stage 4. So early stages, when the rods are gone, you can still protect the cones from dying. And there are many trials now, phase 3, using N-acetylcysteine. There are some other upcoming trials using serine, and serine trophic factor has been used. And the only randomized clinical trial is on vitamin A. They got to phase 3. When all the cells are gone, the electronic retina, Medicare cover the cost of electronic retina. And then at the advanced stage, the many stem cell replacement trials ongoing. And this advanced stage, of course, is optogenetics also will play a big role. This is one of my patients with electronic retina, the first generation, with about 16 dial on stimulating the ganglion cells on top of the retina. Later CMS Medicare approved a 64 dial with a higher resolution. And then many embryonic stem cells on the induced pluripotent stem cell trial ongoing for retinitis pigmentosa and macular degeneration. The challenge for cell replacement is that, first of all, you can see the surgery is kind of challenging. You can see the bubble when the cells are injected underneath the retina. When the cells are placed initially, the white dots is when the patient can fixate, so cell replacement can work. But eventually, you can see the dots start to scatter. The patient are not able to fixate on the graph. The graph is a darker cellular structure underneath the retina because of rejection. Optogenetics will not have this problem of rejection. And conventionally, optogenetics is used to make the remaining cone cells more robust. But then the MCO-010 have the capability not only work on stage 3 when there's still remaining cone cells, as long as the bipolar cells still well connected, and also stage 4 when the cone cells are gone, but the bipolar cells here later are still present, and that's where the expression of the MCO-010 goes. So really can overcome some of the barriers for retinitis pigmentosa to improve the quality of life, because they work at pretty advanced stages, and it does not need to be so mutation specific and can cover all these 80 different genes causing retinitis pigmentosa. If you're interested to read more about precision medicine, gene-specific treatment, CRISPR genome engineering, stem cells, feel free to read more of these books. Thank you.
Thank you, Dr. Sang, for sharing the epidemiology of IRDs, the lack of approved and effective treatments, and the various approaches seeking to bring treatments to patients. I'm going to present our solution to this problem, first starting with mechanism of action of gene-agnostic MCO optogenetic vision restoration with bipolar cell-specific targeting. Next, I'll talk about unique structure and functional characteristic of MCO platform and proprietary AAV-based targeting of bipolar cells leading to vision restoration at ambient light level. I will conclude highlighting the differential product profile that our approach presents for real-world vision restoration. The challenge of restoring vision in these patients is grand because there are hundreds of IRD causing mutations, and each patient's mutation would have to be known prior to photoreceptor loss for classical gene therapy to be effective. And even if confirmatory genetic testing could be completed for each patient prior to significant vision loss, a toolbox of hundreds of gene replacing and editing therapies would be needed to address majority of these patients. Such a toolbox would take decades to develop, while optogenetic has the potential to address these patients even after the photoreceptors or RPE cells are lost. Intravitreal injection that we in our program allows pan-retinal MCO expression needed to re-photosensitizing the retina to have peripheral visual field needed in the severe patient population. Further, the intravitreal injection can be performed in office setting and is scalable to address the widespread need for treatment of these patients. MCO sensitization of bipolar cells, which are proximal to otherwise present rods and cones, creates the potential not just for vision restoration, but also restoration of high quality functional vision, as bipolar cells preserve a lot of visual processing circuitry. MCO10 comprises of an engineered AAV2 capsid, which has been well studied in preclinical and clinical trials to allow expression in bipolar cells. The multi-characteristic opsin MCO is the transgene, which is driven by a mGlu-R6 promoter enhancer for on-bipolar cell specific expression. MCO is a fusion protein that is highly light sensitive across the entire color bandwidth of visual light with fast kinetics to minimize blur. The unique MCO construct maximizes conversion of light to electrical signal, potentially enabling detailed and high quality vision without the need of any external device such as goggles. Our design of optogenetic program combined the important properties required to restore return to life vision in these IRD patients. That is fast and sensitivity across the entire color bandwidth of visible light with fast kinetics. Here you can see the data from our MCO research illustrating the performance of MCO in these key categories. As you can see, the on and off response of MCO is in millisecond range, which is activable by broadband ambient light and sensitive to low light intensity without requiring an external device. So this has the potential to restore high quality vision across severe IRD patients. Intravitreal injection of MCO10 has shown dose-dependent MCO expression in bipolar cells in multiple species. Robust and long-lasting expression of MCO has been observed. Here you can see co-localization of the red fluorescence of MCO reporter M-cherry with green fluorescence of bipolar cell marker in both blind mice and blind dogs. MCO expression could be achieved in 80% of the targeted cell upon intravitreal injection of MCO10. Functionally, MCO10 has shown to restore ambient light perception that was longitudinally measured by visual water maze performance. In severe retinal degeneration mouse model RD10, this example is shown here, where the mice have relatively normal vision in the first few weeks of life when they can be trained to find a platform located under a beam of light. As the degeneration progresses, the mice makes errors and time to reach the platform increases, as you see in the bar graph. Following intravitreal injection of MCO10, the vision-guided performance improved substantially, indicating vision restoration at ambient light level. Notably, this effect was not only significant in mice model of RP, as you are seeing in the middle panel, but also for mice model of Stargardt, where the photoreceptors have mutation, ABCA4 mutation. So both the time to reach platform and number of errors is significantly reduced in multiple animal model, showing the potential of this technology in across different IRDs. Next, in addition to ambient light perception, the MCO10 treatment of retinal degenerated mice led to increased visual performance as measured by optomotor response. So when black and white stripes of different width are moved, the mice move their head, and the improved response of this mice movement was observed longitudinally at multiple levels of light intensity, demonstrating the potential of MCO10 in restoring high quality vision. Now I would like to discuss the key differentiation of our MCO10 program. There have been several attempts to leverage the gene agnostic nature of optogenetics to develop treatment for IRD patient population. A few of these approaches have been able to impart some light sensitivity in preclinical and early clinical studies. However, most, if not all of these approaches have struggled to achieve the necessary sensitivity with the required spectral bandwidth and response time to produce the level of vision necessary to patients to return to life. This slide shows our assessment of MCO10 compared to other optogenetic approaches that we are aware of. Since our approach allows expression of MCO in post-mitotic cells, a single intravitreal injection is deemed adequate for long-lasting effect. MCO10 targets retinal bipolar cells that are abundant and close to otherwise present rods and cones. Thus, MCO sensitization of bipolar cells preserve most of the visual processing. MCO has high light sensitivity and no goggles or external device for amplifying the light are required. Uniquely, due to multiple light sensing components in the fused MCO protein, it is sensitive across entire spectrum of visible light. Also, due to fast response speed of MCO, it has potential for high quality vision without blur. MCO10 has completed phase 1-2 study with positive proof of concept data now available, and in 2 late-stage fully enrolled clinical trial with top-line data expected in Q1 of 2023. The rapid enrollment of all 3 clinical studies demonstrate the unmet need, the innovative MCO platform, and the excitement of the clinicians, as well as our passionate and experienced clinical team to rapidly translate this technology to patients. Now let me turn over to Dr. Aaron Osborne, CMO of Nanoscope, to take us through MCO10 clinical trials. Thank you.
Thank you, Samar. My name is Aaron Osborne. I'm the Chief Medical Officer at Nanoscope Therapeutics. Today, I'm going to present the MCO10 clinical program, which consists of a completed phase 1-2 study in retinitis pigmentosa and the ongoing phase 2b and phase 2 studies in retinitis pigmentosa and in Stargardt disease. I'm first going to talk about the phase 1-2 clinical trial. This was a first-in-human study that was in patients with advanced vision loss due to a clinical diagnosis of retinitis pigmentosa. Participants could have any genotype. Indeed, genotyping was not performed for eligibility criteria. Initially, 3 patients received the lower dose of 0.6 times 10 to the 11. Once initial safety was observed, 3 patients received the higher dose, and then the study was expanded with a further 5 patients receiving the higher dose. The core study was 52 weeks, with the primary endpoint being safety, and secondary endpoints being long-term outcomes, including vision-guided mobility, visual acuity, and patient-reported outcomes. The safety profile was favorable, with no serious adverse events or systemic adverse events related to MCO10 observed. There was no endophthalmitis, retinitis, choroiditis, or any severe ocular adverse event. There was some mild to moderate inflammation in some subjects, which was typically self-resolving, although steroid eye drops were given to 2 of the 11 subjects. Intraocular pressure increase was treated and resolved with IOP lowering agents, and no increase in neutralizing antibodies against AAV2 was observed in the serum, suggesting no systemic immune response. By week 52, there were no signs of active inflammation in any patients, and no patients were taking any concomitant medications. So a favorable safety profile was observed, warranting further study. 2 vision-guided mobility tests were performed in this study. This video of the Y test shows the patient unable to see and move towards the lighted panel. Here the patient moves to the right and apparently does not see the panel. However, 16 weeks after MCO10 treatment, the same patient appears to be able to observe the lighted panel and moves towards that panel and touches that panel. This is an example of an improvement in this Y test score from baseline following administration of MCO10. Overall, we saw improvements in multiple aspects of vision-guided mobility following MCO10. Importantly, MCO10 patients were often able to see and to navigate in dimmer light conditions than they could prior to treatment. Overall, Y mobility test improved over time. Additionally, the A mobility test, which was a similar test but simulated a doorway, showed that the time to find the lighted panel and the overall A mobility test score improving over 52 weeks. The improvements were typically seen around the week 6 to 24 period, and they were fully maintained through to week 52. The diagram on the right shows that all patients that were able to improve their A-test score by 2 light levels did so in this study. So 5 of 11 patients had a potentially clinically significant 2 light level improvement in their ability to perform these vision-guided mobility tasks. Following MCO10 treatment, there were also substantial improvements in visual acuity. Here we see that 7 of the 11 patients improved by at least 15 letters in terms of their visual acuity. 15 letters corresponds to 3 lines on a sight chart and is well known to represent a significant improvement, a clinically significant improvement. The natural history for these patients is not one of improvement, it's one of gradual decline. Besides 7 patients improving by a clinically relevant amount, 3 of those patients actually experienced at least double that improvement. And these gains correlated with improvements in terms of the Y and the A mobility test, as well as patient-reported outcomes. To summarize, there were substantial improvements in visual acuity and in vision-guided mobility after MCO10 treatment that were fully maintained through 52 weeks. Patient satisfaction was high, with reports of improvements in ability to perform activities of daily living and improvements in quality of life. These were reflected by the substantial improvements in the NEI VFQ-25 score that were observed. The overall average composite score improved by over 30 points, and the vision-specific dependency score improved by over 40 points. Clearly, the data from this first clinical study of MCO10 are exciting and support conducting further clinical trials. We've prepared and submitted a manuscript for peer review. The phase 2b RESTORE randomized control trial is a robustly designed, sham-controlled, multi-dose, multi-center trial in patients with severe sight impairment due to retinitis pigmentosa. These patients have visual acuity similar, although some slightly better than the patients in the phase 1/2 trial, which were counting fingers or light perception vision. These patients in this trial have a similar level of vision impairment of 1.9 logMAR or worse. The key eligibility criterion is the presence of inner retinal cells on OCT, which can be assessed by a simple eye scan. The key outcomes in this trial are the multi-luminance Y mobility test, which is a development of the Y test and the A test that were used in the phase 1/2 trial, that was developed after discussion with regulators and other experts, and led to the adding in of obstacles and additional luminance levels to increase the reliability and sensitivity of the test. Additional endpoints are the safety profile, visual acuity, another proprietary test, the low vision multi-parameter test, which now incorporates a 3-dimensional component to simulate activities of near daily living. We also have pupillometry and patient-reported outcomes. The primary endpoint is at 1 year. Starlight is an ongoing open-label phase 2 study in patients who have a clinical or genetic diagnosis of Stargardt disease. This study utilizes a single higher dose of MCO10 and also allows patients with somewhat better visual acuity, although still in the range of severe sight impairment of 1.3 to 1.9 logMAR in the study eye. This enables the use of a conventional sight chart, and that is really the key difference in the assessments, which are otherwise all the same as the ongoing RESTORE study. Both the RESTORE phase 2b and the Starlight phase 2 are fully enrolled. These studies enrolled very rapidly, which we believe is due to the fact that MCO10 is genotype agnostic, so no specific gene mutations are required, simply a diagnosis of severe vision impairment due to one of the diseases under study. There's a lack of alternative treatment options for these patients with severe vision loss that has the potential to restore vision. I think we found that our investigators had a positive first experience with both the trials and the investigational medical product, which led to rapid enrollment timelines. Overall, these trials remain fully enrolled and in the follow-up period. There have been no ocular SAEs reported to date, and we have top-line data from each of these studies that is expected towards the end of Q1 2023. I'm now going to hand over to Victor Gonzalez, who is a retina specialist and an investigator in both the RESTORE and the STARLIGHT study. He's going to share more information on the generation and design of the endpoints that are used in these studies, as well as share his perspectives on MCO10 treatment. Victor?
Thank you, Aaron. Now I'd like to take this opportunity to provide you with some real-world clinical experience with the MCO10. As you know, these patients have severe vision loss, and we've designed two different protocols to evaluate these patients. The RESTORE trial randomizes patients with severe vision loss as a result of retinitis pigmentosa into one of three arms, a control, a low dose, and a high dose. And these patients are then followed for their primary outcome at 52 weeks. The inclusion requirements are that they have severe vision loss, and anatomically they need to have retinal tissue that is our target, which is the bipolar cells. They are then evaluated both at baseline using the Y mobility test, which is a very important test that puts them in real life situations to evaluate these patients' ability to pass these tests at the beginning of the trial, and then they're evaluated through the 52 weeks to see if there's any improvement there. They're also required to pass a 3D dimensional test. It's called the multi-parameter test, and I'll describe it a little later. And then we will evaluate visual acuity for those patients as well. The second phase to trial is the STARLIGHT, and here we have a first in human study looking at patients with severe vision loss as a result of Stargardt's. Now, these patients have a little better visual acuity than the RP patients, and they are given the high dose. It's an open label study, given the high dose of the drug and evaluated through the 48 weeks to see what improvements we have on the patient's ability to function. The recruitment for these trials has been very smooth. And I believe this is a reflection of the fact that there is a large population out there with severe vision loss as a result of these two conditions that satisfy the entry requirements for this study. The fact that for retinitis pigmentosa, this is a genetic or genome agnostic requirement is very important. Previous studies and previous treatments required very specific genetic defects, and that's very difficult because very few of those patients would satisfy those requirements. With this new approach, we can provide treatment for the majority of the patients that present with the severe vision loss as a result of these two conditions. They also receive a very convenient and well-known treatment, a single intravitreal injection. And in order to activate the system, one does not require goggles like in many of the other studies. Just simple ambient light is enough to activate the system. The only requirement, as I mentioned, is the presence of the inner retinal layer. And as I pointed out, a large percentage of the patients that we screen in our trials, satisfied this requirement. For that reason, we had a very low screen failure rate. And I believe this is one of the important reasons why there was rapid enrollment in both the RESTORE and the STARLIGHT clinical trials. The benefit identified by the patients is also the reason why we've had a very high retention rate in all these patients on follow-up. Now, the benefit of having a well-established delivery system is going to be very beneficial, not only for the patients, because a lot of the patients have heard of intravitreal injections, But the benefit also will be that the majority of the retinal specialists worldwide are already experts at intravitreal injections. And this is a very well-established method to treat and deliver drugs. And it's highly accepted by the retina community, and has been a very safe treatment approach for many retinal diseases. The multiluminescence wide mobility test is very important in that it's a situation that provides us with real life activities that the patients need to be able to do successfully in order to have a meaningful and productive life. What happens in this is that we test these patients at different intensity of light. They are required to not only find the illuminated panel, as you can see in this example, but they are required to avoid the obstacles on either side and a central obstacle. So if you see all the red lines here are failures, the patient has to have a very specific approach, avoiding the side obstacles, avoiding the central obstacle, and also being able to identify on which side of the room the lit panel is in. So it's very challenging, and to see the patients in these trials go from a situation where they can't pass this to, at the end of the trial, be able to navigate this has been very exciting. The patients pass if they can have an accuracy of greater than 50%. The patients fail if they touch the LED in a period longer than 60 seconds. And the subjects will pass a light illumination level only if they pass 3 out of the 3 trials. So it's a very rigid, a very difficult test that if the patients are successful, clearly demonstrates a significant improvement in their ability to be mobile and independent. Now, why is this test so important? Well, a change in vision guided mobility by at least 2 illuminations compared to baseline may be considered clinically meaningful. The scoring system enables simple comparison to baseline and or placebo, and is performed by a central mass grader. So it's not us that are doing this. Someone who's completely blind to the treatment is able to evaluate the patients at these different light settings, and will grade the improvement that they see over time in these patients. And why these different illuminations is important, again, as I pointed out earlier, these are situations where the patients will need to face and successfully in real life daily activities, including, as pointed out here, different moonlight nights, to being in a train station and navigating from one part of the station to another. We also will test them using a 3D discrimination system. As you can see here, the patients are asked to identify different shapes. They are tested at 5 different illumination levels, and the patients pass each illumination level only if they get 3 out of the 3 trials. So again, very rigid, very specific change and identification that the patients need to do in order for them to successfully move to the next level. Again, demonstrating the ability of this treatment to improve the function of these patients in real life settings. So in summary, the benefit of the MC10 is that it is a gene agnostic approach to treat a large population of patients with severe vision loss to retinitis pigmentosa and Stargardt's. The multiluminescence wide mobility test and the 3D shape discrimination test can serve as reliable and relevant methods for assessing functional vision in low vision subjects, reflecting the real life patient activities of daily living. The high acceptance of the delivered procedure and the high patient satisfaction have resulted in 100% patient retention in both RESTORE and the STARLIGHT studies. The top line data from the double masked, sham-controlled, randomized, multi-center phase 2b trial for retinitis pigmentosa, or the RESTORE trial, will be expected in the first quarter of 2023. And we're very excited because the impact that this will have on patients with this severe vision loss worldwide is going to be very meaningful. Thank you for your attention.
Thank you, Dr. Gonzalez. So could you describe a little bit about the level of vision impairment that patients entering these MCO clinical studies have got? How does it affect their activities of daily living and their lives in general?
Well, retinitis pigmentosa and Stargardt's are both inherited retinal diseases, and we believe there's a pre-programmed death of cells, and these cells are very important because they're the cells that capture the light. We need these cells in order to activate the neural network from the retina to the brain in order to process images. So it's a progressive, chronic loss of vision to the point where, for example, retinitis pigmentosa patients begin to have severe constriction of their visual field. And in a sense, to simulate that, if you wanted to do it, you can just, put your hands in front of you and have these two little straws looking out and trying to maneuver. So as they progress, they begin to become disabled with daily activities.
And what's their prognosis? Does it continue to get worse through their lives?
Yes, for both it gets worse. For Stargardt's, they tend to start losing their central vision. So Stargardt's patients will lose their ability to read and have acuity. Our retinitis pigmentosa may have that also, but the most common thing to happen is for them to begin to lose their sight vision. So eventually they can meet what is called visually impaired or permanently disabled. And that's defined by the best corrected visual acuity being in the best eye 2200 or worse, and a visual field constriction of less than 10 degrees. And a large percentage of these patients eventually become disabled.
And what's their experience with receiving MCO therapy in the clinic? How do they receive it, and what's it like for them? And what's their experience after receiving the therapy as well?
Well, this is exciting for not only the patients, of course, but also us as physicians, because the natural and normal thing for us to do for these patients is to keep telling them everything's stable as their visual acuity and their function continues to worsen. Given the ability now to provide them with treatment has been exciting. The excitement here is that there's now the ability for us to begin to treat patients agnostic to what the genetic defect is. We've had one treatment before, but it has to be very specific. Now we have an opportunity to treat all comers.
And do you get a sense then of how meaningful a treatment might be that can restore, some vision, any vision to these patients?
These are patients who, many of them are still young and they're still very active. So they've lost their ability to work and be productive. Many of them become depressed because of this. But now, giving them that glimmer of hope, it's been very exciting for them. One intravitreal injection has resulted in many of these patients in these trials to begin to improve. Their outlook is fantastic. Their ability to be independent has come back. And, that makes us and them feel very happy.
Wonderful. I'm going to bring Dr. Sang in here. And Dr. Sang, I'd love to get your thoughts a little bit on the current treatment landscape for these patients. And also, previous optogenetic approaches. Optogenetics has been around for a little while, and several approaches have not shown clear evidence of efficacy in the clinic. What do you think makes the nanoscope approach different, and what excites you about that?
So the current Medicare, Medicaid approved treatments is precision medicine based. It'll be specific to the gene defect. Nanoscope can overcome that. This precision medicine that it can treat all the different 80 mutations and cause retinitis pigmentosa. And previous optogenetics require the delivery to the ganglion cells, quite similar to the electronic retina. There's some limitations of the electronic retina, epiretinal prosthesis stimulating the ganglion cell. And the narrowscope works on the bipolar. Bipolar has a closer, and still would utilize the remaining retina for the integration of the information. So bipolar is a lot closer to the photoreceptor, is in terms of the wiring. In addition, that the advantage of the narrowscope is that most of the Stargardt patients and retinitis pigmentosa patients, when they come to see me, already not so eligible for gene therapy, conventional gene supplementation therapy, because the cells are not there. So in order for gene therapy to work, the cells need to be there. And there's an ongoing trial with antioxidants. It also require the cells to be to protect the remaining cells. So you need the cells to be there for this kind of neuroprotection type of antioxidants therapy from gene therapy. And in the narrowscope technology, that even though the light sensing neuron photoreceptors are absent in the retinitis pigmentosa or Stargardt, the ganglion cells and also in particular for narrowscope, the bipolar are still working pretty well. So the narrowscope has the ability to reprogram the bipolar cells to function to let them function like light sensing neuron.
So what stage of disease, both in Stargardt and retinitis pigmentosa, do you think a therapy such as the Nanoscope MCO10 approach is likely to give the most benefit to patients?
So pretty advanced stages, right? So previous optogenetics technology, I tried to restore some light sensing activity of the cones that have not completely died away. At least to give them light sensing ability to the disease cone cell. That's one of the common optogenetics platform. And I'll do in the ganglion cell as similar to the epiretinal prosthesis. But so I think there's a special niche to utilize the bipolar that narrowscope is using. So patients don't need to have photoreceptors in principle. And most of the Stargardt patient, when they come to presentation, and also the retinitis pigmentosa patient, they don't have photoreceptors. So that the reprogramming of narrowscope technology on the bipolar has this particular niche to cover, in principle, all different forms of retinal degeneration.
Wonderful. So I'd love to bring in Samar Mohanty, the co-founder and president of Nanoscope here. You know, there's a couple of key design features, I think, of MCO10 that are contributing to its potential here. You know, it's an intravitreal treatment, but it also has a broad spectrum opsin that can operate across all of visible light. It's also highly sensitive and has very fast kinetics. And we're just wondering, you know, what do you think were the key design considerations when you came up with MCO10 that have led to this success, both in animals and now we're seeing in humans, in being able to restore navigational vision? Samar.
Yeah, so definitely first, these patients that we are target patient population having pan-retinal dystrophy across the whole retina. So with an intravitreal injection, our target is how to make sure that this protein expressed in the inner retina with an intravitreal one-time injection. So we have a RepCap modified vector where the opsin is packaged, and it allowed in multiple species more than 80% of the target bipolar cell expression. So that was the important factor. And we have a promoter, mGlu-R6 promoter enhancer, which targets specifically on bipolar cells. Then coming to the MCO Opsin, it's a platform technology. It has 3 different components derived from 3 different microorganisms, and they basically cover the whole sensitivity towards the whole visible spectrum. So one of them is a transmembrane ion channel, and then there is a ligand which is sensitive to complementary color, and an enhancer protein which actually re-emits absorbed light to enhance the overall effectiveness of the protein. So all these features about the Opsin does not compromise the kinetics, has a broadband sensitivity and sensitive to ambient light, makes it very unique, and the intravitreal injection with the proprietary AAV vector.
My understanding is that no other approach has been able to use an Opsin that's sensitive across the whole spectrum of visible light. Is that yours as well? There's no other approach that has done this, and how is it possible?
Yeah, that's correct, because this optogenetics is based on Opsin derived from microorganism, and even our cones are specific to a narrow band of wavelength. So as I mentioned, so what we did is the transmembrane protein, which is sensitive to certain color spectrum narrowband, but the ligand is sensitive to other colors, and it acts as a lever to actually pull the transmembrane and allows ions to flow. So we can use all these different components of this MCO Opsin to be sensitive across the visible spectrum.
Got it. And so bipolar cells obviously connect to both rods and to cones, and they could give both peripheral and sharp central vision potentially. We've seen improvements in both aspects in the clinic. Preclinically, we certainly saw improvements in ability to navigate. What's your sense as to what is most likely to be improved? Is it likely to be, sort of central vision, sharp vision, or is it more likely to be the navigational vision that patients lose, early on in retinitis pigmentosa?
I think definitely navigational vision will improve, but our preclinical study also have shown there is an improvement in visual acuity, significantly improved even at 0.25 cycles per degree for a mouse, which will translate to almost 20/60 like vision if we target the bipolar cells efficiently. Of course, it will depend on how many bipolar cells are transduced in the patient, and also the distribution of the virus or the transduction pattern in the retina.
Now, we heard a lot, I think, about the multiluminance Y mobility test in the presentations. And clearly this has been developed based on the phase 1/2 trial, and then adding in additional luminance levels after discussions with FDA and other stakeholders, as well as additional obstacles. I wanted to bring back in Dr. Gonzalez here, because you've got experience with this test. And one key thing about a primary endpoint is that it should be something that is clinically meaningful and relevant to patients. I wanted to kind of ask what your experience of that endpoint is, and do you think it's kind of reflecting what patients need to do in the real world and is a robust endpoint?
I think the endpoint is very important, and that, when you think about what you need to do to be able to function during the day in different lighting situations, right? And early in the morning, you need to be able to drive and be able to see as the sun rises. That can be challenging even for some of us, and a patient that has inherited retinal disease can do that. The lighting in the room at work, the lighting at home. So, in obstacles in front of you, all of these are addressed with the current test. So, and that's reflected on what I've seen of these patients. It's really exciting for me to see a patient who gets guided in, who at the beginning can't do the obstacle, by the end of the study, be able to walk himself into the clinic, go room to room, and be able to navigate that particular, obstacle course. It's very meaningful for these patients, because they will tell you that they're independent and they become much more functional.
Wow. So is that what excites you the most about this approach, that it can bring back that degree of functionality and independence to patients potentially?
You know, these patients, you know, their mood, you know, they're depressed at the beginning. At the end of the study, they're completely different people, have become very productive. Some of them have kept logs, you know, and one of them that was really, you know, tear-jerking in a sense was a woman who basically said, look, I hadn't seen my child's face in over 6 years. And yesterday I saw him for the first time. And I can tell you that that was a tearjerker for my staff. We were all very emotional, very excited to be able to provide that type of recovery for our patients.
That is fantastic to hear. So, you know, if MCO10 were to be commercially available, how many of your advanced retinitis pigmentosa and Stargardt patients do you think it would be a good therapy for?
I'll tell you, right, you know, in our study, you know, right now we've screened, and of all the patients that we've screened, almost, I'd say 80+% of the ones that we screened have been eligible for the trial. So I can tell you that because it's gene agnostic, all we need to make sure of, as Dr. Zhang pointed out, is that the structures that we're targeting are intact, and we've been able to have a high incidence of patients being eligible for the study. And the benefit of the study is obviously seen and appreciated by the patients, because once they're in the trial, our retention rate has been 100%.
And one more question now. We enrolled those studies quickly, and I think I shared a few of my own thoughts as to why that was. What are your thoughts about the speed of the enrollment in these trials and what that suggests with regards to the applicability of MCO10?
Well, there are a couple of things. Some are not great, some are good. You know, the not great one is that there's so many patients out there that need this treatment. That's why there's so many out there that are potential candidates. The good part is that it's gene agnostic, so that we can provide this treatment regardless of the genetic defect. So it's very promising, and I think we will have a very significant impact on these conditions worldwide.
Wonderful. Well, we are approaching the top of the hour. I'm sorry that we will not get to all of your questions. If any of the call participants have any additional questions, please email them to nanoscope@argopartners.com and we'll respond individually. I do want to thank Samar, Stephen, and Victor for a robust discussion and for providing your valuable insights today. It was extremely helpful to hear your perspectives on how the current therapies are limited and how MCO10 can alter the treatment paradigm for both of these diseases and provide a meaningful difference to patients. So thank you again for your participation. You may now disconnect.